induced pluripotent stem cells, and what are the ethics which are related to stem cells. So when I got very attracted towards stem cell research, it was around 2004, and there was one article in Science Daily which reported about the use of embryonic stem cells. That time, adult stem cells were only restricted to cancer therapies, not into some other degenerative diseases. So people were working on embryonic stem cells because they were the most pluripotent or most powerful cells. So this is at the back of a nude mice. The nude mice is devoid of any hair, and you can see this is the back, tail, and this is the 2 ears. So a single embryonic stem cell was injected at the back of the mice. After a few weeks, scientists, they observed generation of hair follicle, skin cells, and sebaceous gland. These all three are the part of the skin. So you can see the single cell has the potential to develop into a particular lineages which is required at that site. It did not develop into bone or any other tissue. So this just highlights a single stem cells. We always correlate stem cells with regeneration. So we just go back and say that is regeneration occurring in nature, or just we are just forcing the use of stem cells for regeneration. So when we see in nature, there are plenty of examples which shows yes, regeneration do occur in nature. There are the few organisms, including the embryo, which can regenerate. But these are the lower organisms. But when we go into the higher complexity of the organism, there's an inverse relationship. That means more higher the organism, the degree of regeneration is reduced. So more examples. Tremble in 1740 showed that hydra, if cut into 2 pieces, can regenerate. And likewise, Aristotle, in much way back, shows that salamander can also regenerate. So in human being, let us concentrate. Regeneration do occur, but there is a various degree of regeneration. So I have categorized 3 points: high, moderate, and low. We all know there's a high degree of regeneration in skin, intestine, and liver, and bone marrow and skeletal muscles, they have moderate and very low in these organs. So why it is important? Let us first think for a moment if there is no regeneration. So there are so many unmet medical needs which are due to degeneration of certain type of cells or the tissue. I have just given 3 examples because these are the, according to me, are the most burden-forming diseases in today's lifetime, because medical science has progressed so much, the life expectancy of human being has increased. Along with that, there are a lot of degenerative changes also. So in these, there are particular type of cells or tissue which are dead. So what we need to restore these cells or tissue. We also know that current drug can only come up to a certain level. After that, we really cannot say that they can cure the disease. So what are the alternatives left to us? We all know, we talk about a lot about organ transplantation, both from liver, kidney, cornea, all these things. We all know there's a shortage of donors. People don't come forward to donate the organs. Of course, it is very expensive and it might not be applicable to too many diseases. Right now, we cannot talk about the whole brain transplant or all other things. So here comes the role of stem cells. Can it augment the current therapy to improve the lifestyles, the mortality, and the morbidity as a cellular therapy? So we all know that stem cell is not new, although recently we have received, 2 scientists have received the Nobel Prizes. But in 1990, Sir Donald Thomas received a Nobel Prize for his use of bone marrow-derived stem cells for the treatment of blood cancer, that is leukemia. This was his paper. He started with animal studies, and now we know that this type of treatment has really become a boon to the cancer patients, and they can be called cured by the disease. I think Professor Bajaj has very well elucidated that the mixture of old and new in this 2012 Nobel Prize in Physiology. Let's just focus 2-3 slides on properties of stem cells. They can renew. Everybody knows that they can go on renewing themselves for a longer period of time, but how do they do it? It's by asymmetric division. Normally what happens when a cell divides, it'll divide into 2 identical daughter cells and they differentiate. But stem cells are really smart. What does a stem cell do? It divides into 2, but it retains a copy of itself, so that this is again a stem cell which can repopulate the stem cell pool, not to diminish the pool of stem cells. And the other daughter cells, they differentiate into the required cell types. Second is telomeric length. What is telomere? All the chromosomes at their end contain a small piece of DNA which is required for the cell to maintain into repeated cell cycle. That means if this length is there, the cell will not die, it will not go out of the cell cycle, the cell will continue to divide for a longer period of time. So stem cells, they have this enzyme called telomerase, so they keep on making this telomere length to particular level. But adult cells, they have very small telomere length. After certain times, like 1 month or 2 months, any other differentiated cells will die, but stem cell will keep on renewing it themselves. For the discovery of telomerase, these three, they won the Nobel Prize in 2009. So everybody knows about the type of stem cells. I just want to highlight that earlier people knew about embryonic stem cells. They are the most powerful one. But now there are fetal stem cells derived from cord blood, cord, and adult stem cells. The list of adult stem cells is growing day by day because every type of tissue in our body harbors stem cells. This is the gift of nature to us. This is the list. Some of the examples are here: bone, skin, neurons, everything, adipose tissue, dental pulp, cardiac, pancreatic, liver, and intestine. And this list is growing every day. So when we initiated stem cell studies and trials in AIIMS, we hooked upon some of the studies. This was a study in which stem cells were injected into the heart, and which showed that there's some benefit in cardiac infarct patient. So I would like to highlight this paper, which I really like, published in Circulation. Here, there were 4 females, a patient of multiple myeloma. They received bone marrow transplantation from sex mismatch. That means they were females, they received bone marrow from a male person. So somehow they had some adverse event, and after receiving the bone marrow, after a few years, they died. Their heart sample was biopsied, and they were screened for the presence of Y chromosomes. This was just part of the study. And the researcher observed that these females have the presence of Y chromosome. This is the light blue dot is the Y chromosome, the dark blue is the nucleus, and this is the cardiomyocytes, the red one. So how come a female having a Y chromosomes? So it was postulated when they received the sex mismatch bone marrow stem cells, and these 4 females were having cardiac problem. Somehow these cells, when given into the infusion, they knew that they have to reach some of the organs which needs them. So the heart was not functioning proper, they were having some problem. So these cells also travel to the heart and reside there, transform themselves into cardiomyocytes. That's why the presence of Y chromosome. And it was not 0.1% or 2%, it was around 23% of cardiomyocytes had Y chromosomes. So this shows that stem cells do their work. But let's see, how do they work? Nobody has seen that how they transform into cardiomyocytes. What do they do? We only have theories to postulate that how stem cells work. First is they differentiate themselves. They reach the site, they differentiate into type of the cells they are required. If you inject them into the skin, they will turn into skin likewise. It is also said that they fuse. They fuse with the resident cells which are present there, cells which is dying or on the verge of dying, and make it again revive itself and do its function. So it is called the fusion of the stem cells with the native cells present at that site. Second is immunomodulation. They secrete some of the factors which immunomodulate, which change the direction and the dying cells, like apoptosis or anything else, which makes the cells not degenerate and revive there. There are certain paracrine effects. This is done through paracrine. This is called releasing of the growth factors. Stem cells have plenty of growth factors. I cannot list it here, which are beneficial for the revival and sustenance of the cells. And of course, they have some angiogenic factors. That means they can lead to more blood vessel formations through these effects. Now, what is a molecular mechanism? So how does it do? So I have just made a cartoon to simplify the process of mechanism of differentiation. For example, this is a cell. So it will get a clue or signal from outside, okay, that you have to change. This signal can be in form of some synthetic molecule or growth factors or cytokines. Then there will be certain gene activation. If we give one factor which is only related to cardiomyocytes, only cardiac myocytes specific genes will be activated, not all the genes will be activated. After that, we all know when genes is activated, you have protein formation. Protein will be formed, and this protein will help these cells to change its shape. It will start differentiating and make into a differentiated cells. This is a very simplified process. So I'll just show you another cartoon. FGF2, TGF-beta and BMPs. These are the three molecules which are required for cardiac differentiation. The moment they attach to, this is the cell membrane, this is the cytoplasm inside the cell, this is the nucleus where your DNA is there. The moment these molecules, they touch upon the cell membrane, whole lot of cascades takes place. These are the few pathways, signaling pathway which are activated. They send the signal into the nucleus and they get attached to the nucleus and onto the DNA, and then translation and transcription process starts. Proteins are released and they trigger the formation of cardiomyocytes. So this is one of the examples. I have deleted few other pathways. So I'll just highlight some of the clinical research which we are doing at AIIMS. AIIMS is a center of excellence for stem cell research by the Department of Biotechnology, with a main aim to do basic research, preclinical research in animals, with the aim to translate this research into clinical trials and in future from benchtop to bedside. So the potential use of stem cells, we know, these are the list of the uses which we can have it here, but AIIMS, we have undertaken clinical trials in these diseases. I'll not dwell upon all these, but give you example of one or two. So how did we start it? We had a brainstorming session of disease-wise, that why we want to choose this disease. Do we have enough number of proof of concept is there? When it is established, then we go ahead. All these trials were from bone marrow derived mononuclear cells. They were autologous because they are from the patient's own self. There's no question of tissue rejections, and ethically also we were much in a better position. So this is an example of limb ischemia, where blood circulation to the forelimbs and the lower limbs is restricted. This is pre and post DSA picture. You can see here, after 3 months, the branching of new vessels have come up. Usually these patients have no other options but amputation, but they have shown remarkable improvement, and there's in the test group there's no amputation, and they can really go back and do their normal activity. In retinitis pigmentosa, which is a very hereditary disease, in this also we started autologous. Like there's a question that why you started autologous, because the gene defect will also be there in the stem cells. We heard that stem cells also have paracrine effect, means they release some of the growth factors. We thought why not use the growth factors released by these stem cells and see what improvement they can do in retinitis pigmentosa. This is a ERG electrogram. You can see here pre-treatment, the area and the macular area is totally dark, and after stem cell injection, 1 month, 3 months and 6 months, 3 follow-up, you can see the activity, the electrical signals are there, the activity is seen at the center of the macula. I'm not saying that these patients are dramatically improved, but little improvement in their visual acuity makes them a little bit self-reliant and independent from anybody's help. The study is still ongoing. Some of the translation research which we did in the lab and which had some effect in the clinics also, I want to highlight. First is corneal surface reconstruction. Professor Geeta will be speaking about it in details. But to show you that we take a biopsy sample, grow them in the lab over amniotic membrane, and do the transplant. She was the student who initiated this work. We first characterized them fully in the lab. This is a few picture of limbal stem cell transplantation, before and after. They are all chemical burn patient, and there's improvement in visual acuity. This again another one. Like this can be done if one eye is affected and another eye is normal. But what happened in cases where both eye is affected? We thought oral mucosa is also having the similar properties of ocular surface epithelial cells. We harvested ocular surface stem cells, grew them in the lab and did some of the transplanted. We have done it in 30 cases and showed some improvement in their vision parameter. Second one is like stem cells from the skin and the hair follicle. Skin is the largest organ in our body, and there were few papers that hair follicle also have stem cells. So we took some studies using skin and hair follicle. You can see here skin biopsy samples and the hair follicle taken out from the back of the head. We isolated cells, you can see here, and differentiated into melanocytes. They are positive for specific staining for melanocyte, HMB and S100, both skin and hair follicle. You can see L-DOPA is a stain to show that yes, they are truly functional melanocytes. And these are, of course, gene studies. They were able to differentiate into neurons and keratinocytes. Just skip that. When we thought that why not use hair follicle stem cells for treating vitiligo? The usual mode of treatment of vitiligo is to take a skin biopsy from the thigh and do the skin grafting at that site. But somehow if the vitiligo patch is big, you have to harvest a big chunk of the skin, and it is slightly painful, and also it leaves a scar on the patient's surface. So we took hair follicle stem cells and we transplanted, isolated the outer reach cells which composed of mainly stem cells, and we did the transplantation, and this is published in British Journal of Dermatology. You can see before and after. There were 15 patients. Now they have been followed up for now 2 years, and the vitiligo is stable. And here one is served as a control, and other we transplanted the cells. So some of the basic research, we focus on hematopoietic stem cells from the bone marrow. We also shifted to mesenchymal stem cells because they are much more powerful than mononuclear cells. They are immune competent because HLA-G is not there, and they can really grow to a large number in the lab. So we have worked on mesenchymal stem cells from bone marrow, cord blood, dental pulp, and adipose tissue. We worked on limbal stem cells, I showed you earlier, then hair and skin from the oral mucosa cells. So we have differentiated them into cardiomyocytes, neuronal cells, hepatocytes, limbal stem cells into neurons and epithelial cells, hair follicle again into neuronal, melanocytes, and keratinocytes. So just to show you about dopamine producing neurons, this is the mesenchymal stem cells and how do they change the shape when they are induced into neuronals. You can see here they are getting elongated, and they are acquiring all the neuronal markers, TRIG1, neurofilament, MAP2, and nestin. By gene studies also, they are positive for all neuronal markers. But okay, you can see here how they have changed the shape. Long projections are there. More pictures. We have also set skin and hair follicle, differentiating into neurons, changing the shape. Again, dental pulp derived stem cells, they are also changing into neurons. Okay, fine, we got neurons in the lab, but how do we know that they are truly functional? Functional means they are able to transmit the neurotransmitter, they are able to change their action potential. The moment neurons are excited, there is a depolarization state. So that has to be checked. That means they are functional. If in future, if they are transplanted, they will be able to do their function properly. So we check their function by calcium ion imaging. The red dots denotes the calcium influx inside the cells. Upon excitation, you can see the cells are much more red, as in the beginning they were like this. So there is influx of calcium from outside to inside. That means the membrane of the neurons is active. The next is cardiomyocyte. As you said, mesenchymal stem cells look like this. When we give them the growth factors, you can see here how do they change their shape. And they acquire all cardiac specific related markers. Cardiac troponin I, MSC, light chain and heavy chain, And their gene studies also revealed that they have truly changed into cardiomyocytes. Okay, let's go to the pluripotent stem cells. In the pluripotent stem cells category comes two cells, embryonic stem cells, and second is iPS cells. So here, embryonic stem cells, which have been worked upon from way back, and recently iPS cells. So IPSC technology, now we all know that you can take adult cells, you can have genes OCT4, KLF, SOX2 and c-MYC. As Professor Bajaj said, now new technology is coming to omit c-MYC. c-MYC is associated with cancer, is a cancer related gene, so people have omitted c-MYC and used OCT4, KLF and SOX2 to change these adult cells and reprogram themselves into ES cells. Basically, what are the methods is, we take the help of the virus right now. It can be a lentivirus and a retrovirus. Only the properties of the virus of infect that it can attach to the cells is taken care. And we hook these 4 genes into that virus. The virus attacks the cells. It only remains in the outer surface. These genes enter inside the cells, go into the nucleus, get themselves attached, reprogram the cells, tell it to behave in their own manners like ES cells, and you get these cells. So they are iPS cells. So what we can do with the iPS? It has won a Nobel Prize. That means it must be having some specific purposes. When you have iPS cells in a dish, they can be used for modeling of a disease. You can isolate from any neurological disease or any cardiac diseases, make a model in the dish, in a dish, need not go into the animal studies, because it is very cumbersome to do some of the animal studies. You can test some of the therapies. In these cells, you can have new drug, some steroid or new molecule you want to check. You can check on these disease cells. And of course, in future, you can have disease cell of a patient, correct the disease by using these technique and give the corrected cells back into the patient. So I'll just show you, these are the ES cells in our lab, and they spontaneously differentiate into embryoid bodies, and they can go into, this is neuronal lineage, ES cells, neurons, and like they also spontaneously beat in the culture, they behave like cardiomyocytes. You can see here in dishes, some of the patches of ES cells, they convert themselves into cardiomyocytes and they beat. Now, next is stem cells and tissue engineering. This is also a very challenging and very interesting area with a lot of effects in future. We are doing stem cells using a biomaterial or scaffold, because stem cells are individual cells. You can have them as a paste, or you can have them in a liquid suspension. You can inject them, but it is very difficult for us to carry stem cells, because surgeons and all other clinicians, they want cells to be placed on a solid surface so that you can carry them easily and you can have more number of cells remaining there. So we take the help of biomaterial or scaffold to carry these cells. There's a whole lot of scaffolds you can have of any sizes, any material, any texture strength. So we are working with IIT Delhi for the construction of artificial cornea. We are taking a clue from the nature. So nature has made this cornea a unique tissue in terms of transparency. So how it is able to maintain transparencies is when we go deeper into the cornea, we see that there's an arrangement of collagen fibers which are perpendicular to each other in different layers. The top one are like this, middle one is perpendicular to the top one, and the bottom one is perpendicular to the middle one. So this special arrangement of collagen gives a transparency to this cornea so that light can penetrate. So we are hoping that if we can mimic this, we are making a scaffold using silk, the serine protein in the silk. It is a natural protein. We have combined it with polynepum, which is a temperature sensitive thing. The moment you lower the temperature, it changes its shape. So these lines are the silk protein made on a glass slide. So we have made small lines. This is under high power magnification. So we want to see that if we plate cells here, can we make them arrange in a linear fashion? So this is our arrangement, that we are trying to grow cells between two bridges so that they orient themselves in a particular fashion. So this is some of the picture. The theory behind is, this is a plastic surface, this is the cell, and this is the scaffold which is coated with polynepum. Polynepum, you decrease the temperature, cell sheet will lift itself up in the culture, and we will lift this sheet, put them one over the other, just to have a perfect orientation. This is to just show you, this video is not working, that cells after some time they start lifting up. So we have isolated cells from 3 types of cells present in the cornea: corneal epithelial cells, corneal stromal cells, and corneal endothelial cells. So these cells will be used for making artificial cornea. Second is long bone defect. We have bone bank in AIIMS to provide some small spicules for doing reconstructive surgery. But for long bone defect, if there is a tumor in the bone, a large chunk of bone is dissected out. So we were thinking, why not to have a model using stem cells and scaffold for these type of defects. So we have used a biocomposite using hydroxyapatite, chitosan, and polycaprolactone. So these are the studies. We make different combination of this bioscaffold just to see that they are closer to the strength of the natural bone. So this is the study to know the degradation, that if they are implanted inside the body, they should also degrade slowly or according to the rate required by the clinicians. We have done some animal studies. We have implanted these scaffold along with the stem cells at the ectopic site. You can see here mineralization is there, and after harvesting, we study the X-ray by mineral deposition. You can see 2 weeks to 6 weeks, there is a calcium formation by these mesenchymal stem cells. And we also check histopathologically that there is no abnormal tissue formation or inflammatory cells. Okay, this study is also very important because this was done on spinal cord injury model, right sciatic nerve transected. We did it in rat model. There were 2 groups. One is test and another is control. In test, we did bone marrow stem cell implantation. In the control, only fibrin glue was put, no stem cells. We did harvest the tissue at day 30 and day 60, and we did a panel of investigation, H&E staining, Masson trichome staining, and Luxol fast blue, just to know the nerve, the myelination, and the presence of any inflammatory cells. So what we got, this is the thing, if this is a nerve, we did the transection here. We took 1, 2, 3, 4, 5, 6 samples. G is the site where cells were transplanted. C is 5 centimeter away, and then A is 10 millimeter. So both distal and proximal, we did the sampling. I will not show you everything. Here, this is test and control. What we see here, the axons are densely packed, and there's less degeneration. There is sprouts of axons here. These are NF. NF is as a marker of neurofilament. Your test group, you can see the degree of neurofilament is more as compared to the control. Here axons are haphazardly arranged also. Arrangement is not proper. And axons are thickly myelinated. You can see the ring structure here. So this is only at 30 day, 60 day also we did this, we found the same thing. So next is we have a certain dose of stem cells. What will happen if we increase the dose? So this was the dose increment study in the same group of animals in the same manner. So control group, group 2 where less stem cells were injected, group 3 high dose of. You can see here,
Dr.
of course.
Sujata,
yes, sir.
Your time is up. Please wind up now.
Fine. So this is again a better example of tissue engineering and stem cells. Here the patient was having collapsed trachea. They used a donor's trachea stripped off to the cells and transplanted stem cells over it, and the same trachea was given to the patient. And after 4 months, the doctors observed that this trachea was able to engraft into the donor patient. I'll just skip all these things, just show you one of the video just to show that people are trying to use heart cell as a bioscaffold for study of heart diseases. Here the rat heart is used and stem cell is injected into this heart. This is a scaffold of the heart and it starts beating in a laboratory. There's again a shady side of stem cells. There are so many clinics which are mushrooming and they are doing stem cell therapy according to ICMR DBT guidelines. These are all clinical trials except bone marrow transplantation for cancer and limbal stem cells. There are also international guidelines. There's few unresolved issues are there before we can go into optimize cell-based therapy. We have to really go into it. The last point is, take-home message, it is a very evolving field. There's a lot of potential, but we need to do systematic and in-depth research to make this stem cell research a hope. Again, I would like to thank all my partners here in this study, IIT Delhi and departments of AIIMS and funding agency, and some of the people who really work with me in the lab. Thank you very much.